LSD would be completely inactive if there weren't proteins within the brain for LSD to bind to, and if these proteins were not to translate the information brought by LSD into something the brain actually understands. But how come peyote mescaline, for instance, is quite a weakling relative to LSD, whereas DMT, unlike LSD, stumbles when it comes to tolerance induction? Both questions lie within the realm of pharmacodynamics, and it just so happens pharmacodynamics is what I would like to discuss with you right here in this video.
Yeah, so far we've spoken about the pharmacokinetics. As I said, pharmacokinetics is about the question, it's basically about the journey. How does a drug find its way to the brain and back again and out of the body? And pharmacodynamics is not so much interested in these questions. It rather asks, once the drug is at its target site, so in the brain, for example, what does it do there and how does it do it? Dynamics means something like force, so it's basically the force or the effect of what the pharmacon does. What the drug does to the body is basically the question of pharmacodynamics. And as I understand, most of you who joined today are students of psychology, so probably you have heard or you have seen the synaptic cleft already. Again, we would enlarge a part of the brain very strongly and would find here a neuron and here a neuron. So this is basically the postsynapse and this is the presynapse. Here an action potential would arrive and would make the vesicles basically become one with the presynaptic membrane, and then the content is being released to activate the postsynaptic receptors. This can be ion channels, but it can also be G protein coupled receptors. And LSD would basically we spoke about the blood-brain barrier, so LSD would come out of the vessel into the interstitium and would find its way here into the synaptic cleft and bind the receptors. We can actually think of LSD as speaking multiple languages. As you might know, there are different transmitters within the brain, and transmitters like dopamine, noradrenaline, and serotonin, glutamate, GABA, and so forth can indeed be understood as different languages. So some neurons speak these languages, others would understand them. And LSD, when we stay in this picture, would be speaking multiple languages. The reason for this is here you see the LSD molecule that it actually shares important constituents of the neurotransmitters. So it shares the indole nucleus with serotonin, and it also shares the benzene ring. This is this one. With dopamine and noradrenaline. In addition, they all have their amide chains, so this makes them actually amines. And this is the reason why the brain misconceptualizes LSD as being dopamine or serotonin or noradrenaline. This is how one can imagine it. So LSD would basically prefer receptors that are somewhere within these systems. And this has something to do with the structure similarity. A very important parameter when speaking about drug receptor interaction is the affinity. It's a really, really important parameter. And you will find in the literature two measures of it, KD and KI. They actually mean the same, so you can treat them alike, but they are determined in a different way. KD is a measure of affinity and KI is a measure of affinity. The difference between both is that KD can only be determined if the drug you are using is available as a radioligand. So if some company has produced LSD and attached some radioligand to it, then you can determine the KD. Very often, however, this is not the case. And this is why you have to determine the affinity indirectly by using, for example, radioactively labeled ketanserin and measuring how well LSD is in displacing ketanserin binding to the serotonin 2A receptor, for example. So this is the difference between both. But let's focus at the KD. It is actually quite easy to understand how this is measured. You have a system, you have a brain system, or you have a you have some cells you're working with in your lab, and you would basically increase the concentration of LSD more and more and more and more. And the more you do it, the more the drug would be able to bind to all of the receptors within your system, maybe the serotonin 2A receptor. So at some point, you have so many LSD molecules in the system that all of the receptors are occupied. This is then saturation. And by convention, the concentration of a drug which occupies half of all receptors is the affinity. And you would basically see, okay, this is where is the concentration? In this case, it would be around, I don't know, 2 nanomolar, let's say it's 2 nanomolar. So you would basically have to go to the 50% binding and then look what the concentration is. So this is what affinity means. Affinity is actually the likelihoods that your drug interacts with a given receptor at a given concentration. And this can be done with all kinds of receptors. Here, for example, done by Lichty, a team from Switzerland. We got serotonin receptors, we got noradrenergic receptors, dopamine receptors, and also some transporters. And here you basically see the affinities of LSD for all of these receptors. When it comes to Ki values, it means the lower the value, the higher the affinity. And based on what we heard here, what this basically tells you, if you have a concentration of LSD of 4 nanomolar in the synaptic cleft, 50% of the serotonin 2A receptors are occupied. This is what it means. And these are very important values. We will also come back to them later when we speak about microdosing and the question whether it's actually possible that at such a small dose LSD is able to interact with the receptor. This can actually predict it based on such data. So as I said on the last slide, LSD speaks multiple languages, so it can actually bind to serotonergic, adrenergic, and dopaminergic neurons, receptors. What I usually tell my students is when you look for affinity values, don't just go for one paper. The reason for this is very often a lot of papers determine one and the same phenomenon. So here, for example, if you go to the KI database, which is open access, you can go there yourself. You just need to enter the drug you're interested in, and then you will get such a list. And here is where the affinities are. All of these are all different studies, and they all determine the affinity of LSD for the serotonin 2A receptor. And as you can see, there is actually quite a drastic variation across the study. So it's actually always recommendable to go there and use a median instead of just going for one paper. Also, when you go into this database, you find all kinds of other receptors. As I showed you on the slide before here from the Lichty group, they investigated a certain amount of receptors, but of course there are much more receptors. And here in the KI database, you actually learn that LSD actually interacts with almost all serotonin receptors, and with most of them with quite high affinity. Only the serotonin 3 family not. So it's really primarily serotonergic compound, and that's why we also say it's a serotonergic hallucinogen. But also seeing this, you actually get to see that LSD has quite a lot of effects, and it's actually quite difficult to predict what all of these receptors do, especially, for example, if you chronically take LSD. It's very difficult to predict how they adapt, how they're sensitized, and so forth. The more complex the pharmacodynamics of a drug, the more difficult it's actually to predict something like this. I've mentioned it before, and I guess you also know it. The key receptor for all serotonergic hallucinogens is the serotonin 2A receptor. So if you consume your LSD, this receptor is being occupied, let's say 50 or 60%, and then you have your unusual effects as Alice here in this case. If you pre-treat Alice with ketanserin, and then Alice would also consume LSD, then Alice will be very sad because it realizes that LSD is not active anymore. And this actually has been shown by some groups in Switzerland again and again, also for other psychedelics. And based on this, but also a lot of animal research, we know actually we need the serotonin 2A receptor. This is the primary pharmacodynamic of serotonergic hallucinogens, and the other receptors might flavor the psychedelic experience, but they are not the main driver. Okay, and let's speak a little bit more about drug receptor interaction. Similar as in pharmacokinetics, there are models that describe how a drug would interact with its receptor. And an easy model is the so-called two-state model, in which we believe that when we go into the brain and we think about the serotonin 2A receptor, let's say we are in the frontal cortex. And these are all serotonin 2A receptors. Some of them are inactive in their inactive state. These are these blue, and some of them are actually in their active state. And they can all spontaneously switch. This is the assumption. But when there's no ligand around, the inactive state would predominate. So that means that there's always a basal serotonin 2A activity, even if there's no ligand present. This is the idea. If now LSD comes in, it would preferentially bind the active state, would stabilize it, and so more and more of the receptors would actually become activated. And this would then, compared to the basal output, increase the outputs. And if LSD were a full agonist, then it would basically induce a full effect, but psychedelics are usually not considered full agonists. So the idea is an agonist would basically prefer binding to active states, would stabilize them, and by the time more and more of the receptors would find their way into such an active conformation. This is the two-state model. And the two-state model is relatively simple, but it also helps us to define certain ligand types. So if LSD binds to the receptor, there are certain things it could be there. It could be a full agonist, which means it activates the receptor to the fullest. It could be a partial agonist, which means it only partially activates it. It could be an antagonist, which means it does not really change the basal activity. And it could be an inverse agonist, which means it would reduce the basal activity. We know, however, for psychedelics that they are partial agonists. So this is what is most important when speaking about psychedelics. Serotonin itself is a full agonist. So the endogenous ligand is a full agonist. Serotonin 2A receptor, as all receptors in the brain. I drink something, sorry. It's actually a protein. And a protein is a chain of amino acids. And here you see this chain. The serotonin 2A receptor is composed of 471 amino acids. And each of these letters is basically an amino acid. And this is then, being crouched together basically, and then it forms such a three-dimensional structure. And it's a G protein coupled receptor, which means it binds to G proteins, as you can see here. It's also called a 7-trans membrane receptor, because this amino acid snake basically would travel the membrane 7 times. That's why we call it 7-membrane receptor. So if LSD binds, it would then initiate the downstream events. This is the activation of the phospholipase C. This then cuts off the heads of certain lipids. These heads would then travel down into the cell, into the neuron, would release calcium from the endoplasmic reticulum, and this calcium then can activate calcium-binding proteins and induce all kinds of follow-up effects. There's also another pathway here via, so I said, PSC would cut off the head of the lipids. Of course, something remains here, and this remainder would then basically interact with the protein kinase C, and this then also can induce downstream events. And what is then done really depends on the environment and the overall state of the brain. For example, what could happen is that the neuron gets aroused by this cascade. It could also be that downstream of it, some genes would be expressed so that new proteins are being generated and inserted into the membrane. Another alternative would be that the pseudo architecture of the cell changes and so forth. So this kind of LSD binds to the receptor would then go on and transduce the message into the cell. So this is the interaction that occurs here. And the question, and this refers to what I said before, is it a full or is it a partial agonist? And this can actually be decided based upon how much of this IP3 is being generated or how much of this calcium is being released. This is then an index of how full or partial an agonist is. And again, this can be measured, otherwise we wouldn't know it. And understanding such a measurement is also not so difficult. And in the context of how good a drug can actually activate such downstream event, two additional parameters are important, the potency and the efficacy. So we've already gotten to know affinity, which is the binding likelihood, and potency and efficacy refer to this IP3 accumulation. To which degree does it happen, and at which concentration of the drug does it happen? Here you see examples of 3 drugs. We got serotonin, LSD, and mescaline. Again, we have in our lab, our little our brain tissue or our cells. We would increase the LSD concentration or mescaline concentration more and more and more. And the more the concentration is, the higher the IP3 production. As you can see, serotonin is actually able to induce a maximum effect, so maximum IP3 production. LSD would only induce about, I don't know, 20% of the maximal effect, and mescaline is a little bit better. And this tells us what I mentioned before, that psychedelics are partial agonists, so they are not able to activate this signal cascade as much as serotonin is able to do this. And the potency can actually also be seen here. So you again look at 50%, go all the way to your sigmoid curve, and then go down to the concentration. And this concentration here, let's say it's 0.08 or something. This is then the potency of serotonin to induce this IP production. And this, of course, can also be determined for the other drugs. And as you can see, the potency might be higher. Here, the potency is higher, but the efficacy is lower. And this, of course, has also been done across a lot of studies. Here you see EC50 values of certain drugs and also the efficacy values. As you can see here, LSD is highly potent, which is in line with its affinity. Again, the smaller the value, the more potent the drug is, because you only need a little bit of molecules to actually induce your effects. For mescaline, on the other hand, you need a lot of molecules. So you really need tons of mescaline before it actually activates the serotonin 2A receptor. And this we also know because, I don't know, what was it, 500 milligrams or something of mescaline you need to consume for you to an effect. For LSD, you only need 100 microgram. So this plays a role here. And what I told you about the maximal effect here can also be seen here. So serotonin is usually around 1 or 100, whatever you want to call it. And then mescaline induces only 60% of the maximal response. And LSD is quite weak, actually. It's only a partial agonist and induces only 40% of the maximal response. So this is what potency and efficacy means. Once the drug has bound to the receptor, how much does it change the physiology of the downstream of the cell, and how much molecules do you need for this to happen? Potency and efficacy. Yeah, as I mentioned before, if we go by the study I just mentioned here, this is a study from Newton et al. If we go by these values, we can actually say that LSD is about 5,000 times more potent than mescaline. This, however, does not mean that when you consume LSD that the psychedelic state is more intense or something like this. Mescaline can be massively intense. It just means you need way less, 5,000 times less LSD molecules in the brain for having a psychedelic effect than with mescaline. So this is what potency means. So sometimes you probably hear in the media, oh, LSD is the most potent psychedelic. And this basically refers to this idea you don't need a lot of molecules to have your effect. Yeah, things are usually more complex than simple models. So we spoke about the two-state model. This two-state model has already been reversed. We by now assume that there are actually multiple pre-existing states. Some say that each drug induces a very own conformation of the receptor, and depending on how the drug changes the conformation of the receptor, all kinds of different cascades can be recruited. So it's not only about IP3, which I showed you before. Where is it? Yeah, somewhere around here, I guess. Phospholipase C. It's not only about this, but also a lot of other things are being activated. And the thing is, and this is what is called functional selectivity, there are certain psychedelics which prefer this pathway. And then there are other which prefer this pathway. And there are again other which prefer this pathway. And this is called functional selectivity. And as we will later learn, some people assume that this concept is very important to understand why certain serotonin 2A agonists are actually not psychedelic. This is a paradox we will speak about later. Also, there is the possibility of the serotonin 2A receptor to actually form bigger complexes with other receptors, and this then allows the psychedelic to recruit cascades which are actually not part of the serotonin 2A receptor, but of the other receptor. So as you can see, downstream of the receptor, all kinds of complex things can happen, but having the concept in mind of affinity, potency, efficacy, and functional selectivity will already give you some tools to understand this kind of interaction. Functional selectivity has all kinds of implications. This is actually results from my own research back in the days at the University of Magdeburg. I asked the question, why is it actually that when you repeatedly consume LSD, let's say for 1, 2, 3 days, that at the end of these 3 days, LSD is next to inactive. This is called tolerance. Whereas when you do the same with DMT, there is no tolerance. This is unusual because they are both acting via the same mechanism. And our research indeed points to the direction that the answer for this is that they recruit different cascades downstream of the serotonin 2A receptor. What we, for example, see is that LSD, but also the psychedelic drug DOI, which also induces tolerance, induce very strong internalization of the receptor. What does this mean? Here you see a cell. What you see here in red is the membrane of the cell. If you now apply DOI, you see that a lot of, so it's not the membrane, it's the serotonin 2A receptors that are sitting at the membrane. This is what you see here in red, labeled by an antibody. And if you apply a DOI, then a lot of the receptors are internalized. As you can see, there's a very thick cluster in the middle. It takes about half an hour to have such a cluster. If you do this with LSD, you also find very strong internalization, but DMT actually fails to induce significant internalization. And internalization is a very important process that the more often it occurs, it at some point ends in the receptor being degraded. So it's being destroyed. And it seems that LSD is more quick to initiate this cascade, whereas DMT is not able. And this might be a reason why LSD is strongly associated with tolerance, whereas DMT is not. The question is now, why is this the case? And what we investigated in addition to the internalization is a certain enzyme, which is called phospholipase D. So it's an alternative pathway. And I didn't include the results here, but what our results show that DOI and LSD are actually able to strongly recruit the phospholipase D, but DMT fails. And this enzyme is actually very important to induce the invagination here of the membrane. You see, in order for this receptor to be internalized here, all the way down here, the membrane needs to be invaginated, and this is being done by this enzyme. So speaking about functional selectivity, whether a drug prefers the phospholipase C or the phospholipase D can have quite drastic consequences for phenomena such as tolerance. So there's indeed functional implications of these different events that occur downstream of the serotonin 2A receptor. Okay, the last bit I would like to tell you about pharmacodynamics. Of course, beyond the mere molecular level, we also need to ask, okay, once all of these things go on in the neuron, in the individual neuron, what happens actually to the brain as a whole? And here we actually enter the field of neurobiology. Of course, focusing today on pharmacology, I can only briefly talk about neurobiology, but let's give it a go for 1 or 2 slides, I think. We know that the serotonin 2A receptor is across the whole body. You find it in a lot of regions of the body, but the highest expression is indeed here in the cortex. In the human, you would find it a lot in the frontal cortex, but also here in the visual area. We know from in vitro electrophysiology that if you apply a psychedelic into this region here, the pyramidal cells, which are localized there, the layer 5 pyramidal cells, they increase in excitability. So if you repeatedly stimulate this slice preparation, then normally these cells would habituate, but when LSD is in the game, they don't habituate. So there's an increased excitability and a reduced refractoriness. If we then move further away from these in vitro studies and go into EEG and MEG findings in humans, we know that when a lot of such cells fire together, we have these EEG waves. And here you have 3 different neurons. You see, they all fire in the same pattern. This is called a desynchronization. So the brown one would be the upper one, this would be this one, and this would be this one. All neurons fire in the same way. And if you have hundreds and thousands of this firing in the same way, we would see this in the EEG. If you now apply a psychedelic, what happens is that they totally fall out of sync, which means neurons all of a sudden start to do things that are not in change with the others anymore. And this is called desynchronization. And this is something that really has been highly replicated across studies. So this is really something psychedelics seem to do when you go into individual areas of the cortex. They desynchronize the activity there. If you go beyond local areas, so just beyond local, this would be local. But if you now think of networks, like a network between this and this region and this region, then we normally know from the brain that there's a certain amount of modularity. So there are certain networks, such as the default mode network, which fire and wire together. And then there's another network, which is called the salience network, which was also fire together, but they would usually not interact. If we now give a psychedelic, this modularity is basically eliminated or reduced. As you can see here, is reduced, and these networks would less interact within the module, but all of a sudden start interacting with one another. And this is then interpreted, for example, in the context of ego dissolution. So this is just a very brief idea. All of this is also part of pharmacodynamics. So it's not just about the molecular level, but also these higher level effects. Also, actually, the psychology and the psychological effects, ego dissolution, can also be considered a pharmacodynamic effect on the level of the psyche. This is so we are actually also interested in this kind of research. So at the Institute for Psychopharmacology at the SETI in Mannheim, we also follow up on this kind of research, and we do this in animals. And the problem with EEG in humans is that EEG only gives you an overall output of the cortex as a whole, but you cannot really target the cells you are actually interested in. We are only interested in the serotonin 2A receptors that are, we are only interested in the pyramidal cells that express the serotonin 2A receptor. And this is something you can do in animals. You can also, in animals, go beyond the cortex and see what's going on subcortically, what EEG cannot do. And we can also measure with high resolution transmitter release, for example, glutamate release. All of these things are impossible in humans, and this is the advantage of the methodology we are using, which is called fiber photometry. So we would basically label here, this is basically the cortex of a rat. You see, it's a coronal section of the frontal cortex. And this is here the olfactory nucleus, the piriform cortex, and this is the frontal cortex. And this band, which you see here, this is serotonin 2A receptor expression. And when we have a lot of such sections, we can actually determine the distance from this midline to the serotonin 2A receptor. And then we know that the layer 5, the layer we are interested in, is around 500 micrometer away from the midline. And this is very nice to know this, because then we can directly record from this area. And this is what we did here. This is basically glutamate release in the prelimbic cortex, so the frontal cortex, which is a central region of psychedelic activity. And when we find, if we apply a psychedelic, this is what happens here, the glutamate release is desynchronized. So this is something that could not have been shown with any other methods, could not have been shown with EEG, and this parallels what people know from EEG. What is unknown so far, so this is unknown, of course, but what is also unknown, what subcortical areas do in parallel to this. And we see very often people believe that psychedelics primarily target the cortex, but it's also possible that they actually target certain regions in the subcortical area, and this might then actually have bottom-up effects. So as you can see here, if we apply our psychedelic, the effect here in the subcortical area is much more drastic than it is in the cortex. And this way we can actually expand on the knowledge. So if human research tells us, okay, the cortex desynchronizes, we can actually ask which cells are involved, what neurotransmitters are involved, and yeah, how is the subcortical region perhaps causatively linked to this.
We have now spoken about the two main pillars of pharmacology, namely pharmacokinetics in a former video and pharmacodynamics in this video. There will be a third video in this 3 parts little pharmacology lecture of mine, where we'll speak about hot topics in the field of psychedelic research at the moment, namely microdosing, and the question whether microdosing yields high enough concentrations to activate serotonin 2A receptors. We will speak similarly about endogenous DMT and the question whether there are conditions at which endogenous DMT is in its concentration high enough to be psychedelic, and we will speak about the paradoxical finding that there seem to be serotonin 2A agonists which are not psychedelic. If any of this steals your interest, I will link the videos here at the site, and I would love to see you there, and up till then, take care.